No, it is not identical to last year. There are a few new things that we have added to the tool, and we also just want to, rather than focusing really intensively on all the details of how to use this tool, we're going to talk a little more about interpretation and recommendations for farmers, how to work with farmers using it. And so it will, I think, add some new information over last year's. If this is your first webinar on the green manure bioassay, you may end up with some questions at the end of this or throughout it on some of the details of various aspects. And so I'd encourage you to afterwards go to the Organic Agronomy Training Facebook page, and then last year's webinar is actually posted there, and you can watch it to get some more of the details that we're not covering quite as extensively this time. And if you have trouble finding that, just get in touch with Iris or myself, and we can help you out. Okay, so we want to talk about nutrient management on organic farms, and some of the tools that we've developed here at the University of Manitoba in cooperation with Dalhousie University as well, to help farmers and agronomists make better decisions, more informed decisions on how to manage their soil fertility. And the reason that organic farms are kind of a special case and that we don't just rely on traditional soil tests is that nutrient cycling works differently on organic farms. And so we need tools that can accurately capture what is really going on, because we're relying on green manures and perhaps animal manures or some commercial products that are released very slowly over time, such as rock phosphate, for example. And so it's a little different than conventional farming, where you take your soil test, it tells you how much you need to add, and then you're done. And so these tools, like I said, they have to be well suited to these type of organic fertility management practices, and they have to provide some useful information to farmers and agronomists. Up till now, we've been kind of relying on the principles, the general principles of organic fertility management, without having a lot of specifics to apply in particular situations. And so that's what these tools are about. So the tools we've developed are this green manure bioassay, which we're talking about today. And we've also developed a nutrient budgeting tool, and we're going to do a full webinar on that later on in the fall. I'm also just going to touch on it really briefly for those agronomists who are working with Iris's nutrient management program through POGI. We'll touch on that in a little more detail, just a little bit of detail later on. But you will be getting more information about that nutrient budgeting tool later. And so together those, in combination with soil tests, can provide more information about what's going on in the soil and the crops, and perhaps what can be done about it. So these tools add some precision to just visual observations that we might make on crop performance and that kind of thing. But they are just tools, and so tools are, and oh, there we go.
Leaving conference.
So we still, as agronomists and farmers, we still require good agronomic knowledge to know how to interpret the results of the tools, and also how to move forward with any recommendations. I also want to talk a little bit about how we use these tools together with farmers. And we are really encouraging people to use what we're calling a farm co-design approach, or a cooperative farm design approach with farmers, where the agronomist is not just someone who comes and collects the samples to do the test and hands over the results and end of story, but the agronomist and the farmer actually work together to identify what the farmer's goals are, and how some additional knowledge might feed into the farmer being able to meet those goals more effectively. So I want to give you just an overview of this farm system co-design before we get into the details of the tool itself. Now this is, farm system co-design is not a cookie cutter approach. There's no set recipe or formula for it. This is a plan that we developed for our own process that we use here using these tools, but really a farm system co-design approach can look very different depending on the situation. And this is a type of thing that can almost just happen very naturally, but sometimes it can be helpful to actually map it out like this and see where the interactions are between the different people involved and where certain tools are used and how the decision-making process works. So you can see here we start with collecting information, and that would be from individual fields and farms. The little red people represent the researcher or the consultant or agronomist, and the blue people represent the farmer. And you can see they both have a role in collecting that information. The agronomist might be collecting plant and soil samples, but meanwhile the farmer is also collecting information on yields, quantities of products that they've used, that kind of thing, as well as field observations on what the weeds are, the pests, how the crop looks, and that kind of thing. That information is then gathered up by the agronomist, and here they can use some of these computer-based tools that we've developed to put things together just to manage that data. And those tools are, like I said, the green manure bioassay and the nutrient budget or nutrient balance tool. So collecting that information is what we're mainly going to be talking about today, but we need to be aware that that leads into ultimately a farm plan co-design process where the farmer and the researcher together are identifying the strengths of the system and some key constraints, and ultimately perhaps changing the farming practice. And so you see there's lots of arrows going every which way in this diagram where, you know, a decision made somewhere affects another one somewhere else. You can see after the farming practice may be changed, it goes back up to more collection of information, of field observations, etc. And I'll just mention really briefly here, we can also do, using these tools, we can actually do some scenario development where we would test, you know, so if I were to change my rotation in such a way, what might the results look like? Or if someone is just starting out, what would be an ideal rotation for me to use with the use of how much manure, for example, and then we can create a system that is sort of an ideal for them to shoot for. So that is something that this flow diagram is something that I do refer to sometimes in my own work as I'm trying to think about where we are in the process and where we're going. In more practical terms, the tools that we've developed can be sort of spread out over a timeline like this over one season, where you can see the steps of each tool and how they relate to other sort of general interactions with the farmer. So in spring, we start with an initial contact with the farmer, as well as some observations of crops and that kind of thing, and then get right on to collecting into the green manure bioassay tool. And we'll talk about all these steps in just a moment, but there is some sample collection that happens fairly early in the year because it needs to be at the peak of green manure growth. And so this is a tool that you can have the data back, you know, by early fall, maybe even late summer, and be ready to discuss the results with the farmer fairly early on. And it is important to discuss those results as soon as possible with the farmer because they may actually wish to take some action that fall in terms of perhaps applying manure or some other, or sourcing some other input. You'll see the purple rectangles are the nutrient budget tool, so that happens a little later in the year. And then at the end of the year, over the winter, is when discussion of all the results and discussion of the management options with the farmer can occur. We'll come back to this later, but I just wanted to give you an overview of how those tools fit together into one season of working with a farmer. So let's get into the green manure bioassay itself. We're going to give you just a quick overview of this theory and a summary of the sample collection and processing and analysis, how you do all that, how you compile all that data and get all the results, and then interpret the results. And for the results, we're going to focus on phosphorus because that is the nutrient that is most frequently of concern on organic farms, at least after nitrogen is taken care of. I'll just pause there. If you have any questions, I haven't seen any pop up yet. But if you do have some, please feel free to type them in, and we'll continue on. Okay, seeing none, let's move on. So what are we going to measure in this green manure bioassay? We want to measure the plant biomass production, and we want to know how much of that is a legume, how much is non-legume, and how much is weeds. And you'll see how that information becomes important a little later on. We want to measure the nutrient concentration of that plant material, and we send samples off to the lab for that. We also want to take soil samples because they give us a good comparison to the plant tissue samples, and also provide some additional information that the plant tissue does not provide. And we're also going to take some field observations that provide some really useful data on green manure management and general crop management on the organic farm. And you can keep track of all that data with a spreadsheet tool that we've developed. It was in the email that Iris sent out prior to the webinar. There were actually two versions sent out. One is a blank one, and one is actually filled out with an example. And you'll see in the file names, one ends with the word example. And so you can look at that one, you can play around with it and see how it works before you start working with your own data, if you like. And there's some data sheets in there for writing down information when you're in the field or in your lab, in quotes, whether that's your basement or your garage or your shop or whatever. Sample weights and that sort of thing. And then there's a form for data entry, and it does a whole bunch of automatic calculations for you, and then a summary form in the spreadsheet for compiling the results and that you can print out and give to the farmer. So let's get into the sample collection a little bit. This tool is focused on the green manure crop, and specifically a legume green manure crop. I know the term green manure is used more generally to include all sorts of other crops, but we're really focusing on the legume green manure crop. The reason for that is that a legume green manure should not be limited by nitrogen if it's fixing properly, and so that makes it more sensitive to deficiencies of other nutrients in the soil. So if there is no legume green manure crop, a next best choice of crop would be a grain legume, a pulse crop like peas or lentils or soybeans, or a legume forage like a hay crop, as long as the legume is at least a semi-dominant presence in that mix. The key is to have a legume there providing nitrogen for itself and for any other crops that are in the mix there. The timing of sampling is also fairly important because the results that we get are calibrated for a crop that is in the flowering stage. Nutrient concentrations of crops vary as they mature, and so depending on when a crop is sampled, it can sort of throw the results off a little bit. So the flowering stage is the best stage to be sampling a green manure. That's when the legume is flowering, I'll clarify that. And often that would be as close to the green manure termination date as possible. For one, that's the ideal time to terminate a green manure. but also it's important to keep in mind that people may actually be terminating a green manure early for a variety of reasons, such as if it's a dry zone and they are concerned about conserving soil moisture, or perhaps it was a late seeded green manure and they're getting ready to seed a fall crop like winter wheat or fall rye and just need to terminate it or need to manage weeds or whatever. If the green manure is going to be terminated before it reaches the full flowering stage, then taking those plant samples as close to termination as possible is the way to go. And just to be aware, as I mentioned, that sampling too early or too late can compromise the test results. It doesn't mean that you shouldn't do the test, but it does mean that some care needs to be taken in the interpretation of the results. If you have a farm that has perhaps many green manures or legumes, and you still you have to make a decision about which fields would be best to sample. I would say it is the best bang for your buck to focus on the fields that have been producing the poorest crops or seem to have some sort of problem in them. Or the next best, maybe not next best, but another approach would be if there was a field that was doing poorly and an amendment was applied, to do a follow-up and see what the effect of that amendment was. And I will just mention here that this test is not designed to be used on other types of crops, grain crops, oilseeds, anything like that. It really is designed to be used on a legume. So now just to a bit more of the nitty-gritty of what we will actually be collecting. And along with that, the spreadsheet files that Iris sent out, there is a PDF document that has some detailed instructions, so you can always refer to that for more information. Or, like I said earlier, you can watch last year's webinar and it has some more detail. But when you go out to collect these samples from a field, there's going to be several kind of types of information that you want to collect. First, there's some field observations that we want to collect, and some of that is essential information in that it really is needed for correct interpretation of the test. And that includes what crop it was, the crop stage, as well as legume nodulation, whether the legumes are present and whether they are nice and red or pink inside, like this little photo shows. I realize that some crops have extremely small nodules, and it's not always possible to see the color inside. And so then you have to use your best judgment. Is this a very common legume species like sweet clover? Does it look well nourished and like it's fixing? Then you can probably assume that it is. So there's a little bit of judgment required there. It's also good to collect some supplementary information when you're in the field anyway, and this can help again with interpretation. Simple things like the crop height can help you afterwards when you're looking at all your data you've collected over the year. It can help you sort of calibrate your eye for how much biomass a crop has produced, based kind of on its height and, say, the thickness of the stand. The visual ratings of the percent legume and the non-legume and weeds are, again, it's a good sort of self-calibration exercise to take a visual rating and then also to measure those components later. And then perhaps someday we can all get to the point where we can just do visual ratings. I don't know. I've been trying this for years and I'm still not very good at visual ratings. And I suspect most of us kind of struggle with that. It's also good to take a note of what the weed species are in that field, because that will come into play when you're discussing management with the farmer. And then there are also some samples that are essential to collect. We need to collect crop and weed biomass. That's the above ground plant growth from a known sample area. We'll talk about that in just a little more detail in a second. And we also need to collect soil samples for comparison and for some additional information. In the past, we've been suggesting that you collect both a surface soil, like a 0 to 6 inch sample, as well as a 6 to 24 inch sample. And certainly if you have the time and the tools, that is a good recommendation. If you are lacking time and tools, please at least collect the 0 to 6 inch sample, because that provides the most important information. And provides the most important information for the future management as well as for comparison to this tool. Okay, we've got a quick question from Liz. Do we want the percent by weight or by volume? We are going for percent by weight, and because that's where it all boils down to, is the plant biomass, the weight of the plant material. And you will find that legumes are tend to be lighter than they appear. At least that's my tendency. And then and grasses or other grasses would tend to be a little heavier than they appear in a mix. Next question is, in the case of a multi-species green manure, do we need to collect stage and height of all crops? I would say it is good to take note of the stage of all the crops, and height. You start getting into a lot of measurements if you're going to collect the height of all of them. I typically just take a total height of the tallest species in the mix. And an additional clarification on the weight of the biomass, that is wet weight or dry. We are ultimately aiming for a dry weight, although you'll see in a moment where the wet weight may actually be a good measurement to take too. We'll get to that more in a moment, but ultimately it's the dry weight of the plant material that matters. And I actually had an example of a farmer emailing me wondering if, can I assume that my crop is 20%, 25% dry weight compared to the fresh weight? And I said, no, you can make no assumptions on that, because the moisture content of a green manure can vary widely. And so he kept drying his sample down, drying it in an oven until the weight leveled off. And as it turned out, it was only 8% dry matter compared to his wet sample. So it was a really lush sweet clover crop before flowering. But it just stresses the importance of focusing on the dry weight. Okay, now when you are sampling these green manure crops, it can be challenging, and you go into some of these, for example, a sweet clover crop that's 5 or 6 feet tall with a little round hoop, and you start realizing it's going to be hard to sample. So I talked about this more detail in last year's webinar, but just want to point out that an open quadrat like this is easier to use than a closed one, because you can actually sort of separate the crop and slip it in to the crop. We use these little stickles for cutting them. You may have a tool of choice that works, but these are we find they're really good. You'll never run out of battery power on those, and they have a serrated edge that works really nicely for cutting plant stems. And you'll be cutting these plants as close to the ground as you can to get an idea of the total biomass. Now, another approach that we can use where crops are planted in nice discrete rows that you can find is to actually just put down a meter stick, if you've got a meter stick with you anyway for measuring the height, and cut 2 rows of crop to a certain length, as well as the weeds that are in between those rows. And there's a lot more detail on that in last year's webinar. I'm not going to go into any detail on that right now. So once you collect all this plant material, and I should mention that you should be sampling at usually about 5 places in a field, you are going to end up with a fairly large pile of plant biomass, and we're going to get to how to deal with the volume of that in just a moment. But I want to discuss the steps on what you're going to do with all of that plant biomass first. So we want to know how much of that plant biomass is legume, non-legume, and weeds. And so we are asking that, recommending that you sort it into those components. If you've got a multi-species mixture where there's, say, more than one legume, more than one grass, I personally don't sort into those particular types. I just put all the legumes together, all the non-legumes that are supposed to be there, like the seeded non-legumes, into one pile, and then the weeds into another pile. We're then going to dry them, and air drying them is fine, but we do want to get them actually fairly dry, and that can take a surprising amount of time if you're just air drying them. If air drying, we want, to get to that point where it feels like dry hay, which is assumed to be around a 15% moisture. Or when you send your sample in later, you can actually have a moisture test done on to see what the actual moisture is. If you have an oven available, you can dry them in a low temperature oven. It needs to be at a low temperature to avoid destroying the nitrogen and other nutrients in the sample, but you can actually dry a sample in the oven. We're then going to weigh the sample, record the weight. So a reminder, you'll have a legume sample and a non-legume sample and a weed sample, probably, and so all those weights need to be recorded. And then, after we're done with all of that, we're actually going to put it all back together. I know our summer students hate that when I tell them that now we have to put it all back together after they sorted it all out. But we're going to combine it all back together into one sample per field for the analysis. And we send it off for a nutrient analysis. The key nutrients we want are N, P, K, and S, although we do find that depending on the lab, the cost can be pretty similar to actually just get a complete nutrient concentration analysis compared to just getting those 4 nutrients. A feed analysis will also do the trick. You just have to, it'll report protein instead of nitrogen, so then there's just a conversion that needs to be done there. And again, if you are sending in an air-dried sample, it is a good idea to ask for the moisture content to be analyzed. So we've experienced this ourselves, and we've had some agronomists ask us, I have this mountain of plant biomass. Do I really need to dry this all and send it all in? And so we've developed a bit of a procedure for taking subsamples, just taking a portion of that sample to be dried and weighed. But the key with subsampling is that it needs to be accurate. You can't just grab some and separate it out and weigh it and send it for analysis and hope that it sort of represents your field. That subsample needs to represent the whole sample in quality. So it needs to have the correct proportions of the legume and the non-legume and the weeds and even the types of weeds. And then it also needs to represent the whole sample in terms of quantity. So it needs to be a known fraction of the total. And we also want it to be a manageable size when it's done. So we're targeting a final sample of around ½ pound to 1 pound of dry material, which should be pretty manageable to send in. And so the way we do this is I did a bunch of kind of backwards calculations, so if I want to end up with such and such a size of sample, what would I start with? And so basically what you can do is weigh your whole sample at the beginning, either the whole pile of it before you sort it, or sort out the legume, non-legume, and weeds, and add up those amounts into the total fresh weight. And then if you, depending on what that weight is, then you would choose what we're calling a subsampling factor for how much of that total sample to actually carry on through the rest of the process. So, for example, if you end up with 1,500 grams of material or less, you would just take the whole sample. You wouldn't do any subsampling for it because that whole thing will be probably less than a pound when you're done. Whereas if you've got say 1,700 grams of material or 2,500 grams, we would recommend you take half of it, 0.5 of your sample. Similarly, if you get into larger and larger amounts, if you were using, for example, a really large quadrat or took a lot of samples or had a really productive crop and ended up with a very, very large sample of 10 kilograms of fresh weight, then you would take only 10% or 1/10 of that sample. Now, the key to that then is that this factor applies to each component of your sample. So you would take, let's say we're using this one where we had, for example, 4,000 grams of sample. You would take 30%, 0.3 of the legume weight, and you would take 30% or 0.3 of the non-legume and 30% or 0.3 of the weed. And there's a little tab that we added in the Green Manure Bioassay workbook that helps with the math of all of those to figure out exactly how much of each component you should take. So if you have been struggling with large samples, I encourage you to take a look at that, try it out, see if it works for you. The important part later on then is to remember when you're calculating the total biomass is to multiply it back up so that you end up with the correct total at the end. Any questions about that subsampling? It's a new piece that we just introduced this year, and it is probably the kind of thing that once you actually do it, it'll become a lot more clear, but just wanted to let you know that that is available for you to use. Okay, we'll carry on, and if some questions pop up, we'll deal with them later. So once you have dried and weighed all those samples, then you have some results to deal with. And so in that Excel spreadsheet, there's a tab called Data Entry, and there you can enter all the field observations you took, the crop heights and the percent weeds, legume, non-legume, all of that, as well as the biomass sample weight that you obtained by weighing those samples. And then it helps you out with the math, and it does a whole bunch of automatic calculations with the averages, calculating the total biomass and the proportions of each component. So I want to give you a little look at what that looks like in case you haven't seen it before. It looks like this. You'll see there's lots of room to add some information about the field and the farm and the crop, some notes on the crop if you like, the date that you were there, the size of quadrat you were using, all that information you'll need for your later calculations, and also the nodules and their activity. I'm going to skip down to the bottom here. You'll see there's a place for entering whether the samples were oven dried, your quadrat size, and here you can put in that subsampling factor that you used, if you used one, and then that'll again automatically calculate the right biomass for you. And so the biomass is shown down at the bottom there on an as-is basis as well as on your dry basis, and you also have the percentages of each component. So for example, we have 14% weeds in this one, 48% legume, and 37% non-legume. So that's just a brief overview of what that looks like. I would encourage you to check it out for yourselves and get in touch if you have questions about that. We then go on to the summary tab, which is where all of the key kind of essential results feed into, and where you can also enter the plant nutrient concentrations and do some comparisons to levels that they should be at. We've also, another new thing for this year is we've refined our estimate of nitrogen fixation based on the green manure biomass, and I'll talk about that in a little bit. So this summary tab, I'll show you what that looks like. I have a blank screen. I'm not sure if you're able to see it. Iris, can you let me know whether everyone else is able to see the content of this slide? Okay, it appears that it's good for other people, not good for me. I'm going to look at my PowerPoint and see what I'm apparently showing you. Okay, so this summary tab, again, it'll show you the farm name as well as some of the field information. You'll see those blue cells are actually filled in automatically. All that information just comes automatically from where you entered it on the other tab. And the white cells are where you will enter your own information. And so you can see kind of down in the lower left side of the page, there's a place to enter the soil test results. You just have to get those off of the results from the lab, enter them in there. You would also give the soil test results directly to the farmer, the complete test, but it's just to have them there to side by side with the analysis of the green manure plant tissue for comparison purposes. So, for example, this farm had 13 pounds per acre of nitrogen. Oh, there it showed up on my screen, and 5 parts per million of phosphorus, etc. And you can enter the pH, organic matter, etc. Then, under the green manure crop is where you will actually enter the nutrient content that comes back from the lab analysis of the plant tissue. So here we had 2.6% nitrogen, 0.25% phosphorus, 3% potassium, 0.31% sulfur. Besides those, under critical values, we have indicated some amounts or some levels at which we think, and the literature shows, that that problems begin to develop. I should say when you get below those levels is when it's an indication that there's a soil fertility issue. In particular, we want to focus, in terms of soil fertility, we want to focus on phosphorus, potassium, sulfur. The nitrogen value here has less to do with soil fertility and more to do with how quickly that green manure will actually decompose. So we'll just leave that for the moment and focus on P, K, and S. And so you see here, on this farm, we've got 0.25% phosphorus compared to 0.2% is the critical value. This is greater than 0.2, and so it appears to be okay. Down below that, this is new for this year, we have an estimate of nitrogen fixation, and I'll talk about that more in a little bit. Just wanted to give you an overview of what that looks like. So there we go, the nitrogen fixation. A little bit then about interpreting various aspects of this test. And I hope through this part it'll become clear why we need certain of those field observations that we took. So the total biomass of the green manure we don't have target values for those, but we, generally speaking, more is probably better, but there can be a wide variety of reasons why you might have a low amount. This amount of 33, or basically 3,400 pounds per acre is kind of on the low side from what I would expect, but we also see that this crop was only in the bud stage and was being terminated early for whatever reason. And so that provides some information that would indicate that it's perhaps not a fertility problem that's causing low biomass, but just that the crop was in the early stages of growth. Percent legume and percent weeds. Here we see we've got 37% legume in this example and 18% weeds. That percent legume is a little on the low side. We would aim for usually 50% or more. What is the reason for that? There can be so many reasons that there is too little legume in that mix. One of those might be that it was not inoculated properly. However, we can see that this one had its nodules present and active, and so we can rule out inoculation or lack of nodulation as a problem here. And so then perhaps, you can kind of go down the steps of what additional issues might be, and perhaps it was a seeding, just in the mixture, that the legume seeding rate was kind of on the low side compared to the non-legume. Question about what we would consider an average total biomass. That depends very much on the crop and the stage. We hope to see generally 5,000 pounds per acre or more, although that doesn't always happen. Anything less than 4,000 at the flowering stage, I would say, is a bit iffy. And you'd definitely want to be at least 4,000. Okay, moving along to the crop nutrient concentrations in a little bit more detail. As we said, those are shown there in your summary tab. And there's a few things to consider, like when we were talking earlier about the time of sampling. And so if you find, if you did your sampling when, for example, at the bud stage or a little on the early side, you may find that all those values here are kind of on the high side compared to the critical values. And that's okay. It doesn't matter, but just it's a possible explanation for why those values were kind of high. Similarly, if all the values are kind of on the low side, like near the critical values or just a little below, you have to consider the possibility that maybe the green manure was actually kind of mature, or had been growing very rapidly, and all the nutrients were diluted in the plant. So just to be aware of kind of the overall trends among that whole set of numbers before you start looking at individual numbers. However, if you do look at those numbers and you see that only 1 or 2 are low compared to the critical values, and the others are fine or high, then it would seem that a deficiency is likely. So let's go through an example. So here's farm 1. The soil phosphorus is categorized as low. It's 5 parts per million. The plant phosphorus is 0.25%, which is well above that critical value. And so from that, we would determine that there is no immediate phosphorus deficiency. However, with soil phosphorus at that level, I would be recommending to the farmer that they keep an eye on their crops and watch for any other signs of deficiency, and also probably start scoping out the options for adding a phosphorus source of some sort. Another example, on a different farm here, our soil phosphorus is 4 parts per million, which then crosses that line in at least the AgVise categories down into the very low category. But really not that different than farm 1. However, the plant phosphorus is 0.12%, which is well below the level of 0.2%. And the other values are near or slightly above the critical values for those other nutrients. And so there, I would identify that as having a phosphorus deficiency. So that same process can be followed for the other nutrients, although we don't have, we have more information about phosphorus than we do about potassium and sulfur at this point. So based on those phosphorus concentrations in the plant, we can sort of divide things into different management zones based on those results. And this is actually the data that I've gotten from my own sampling over 2 years, 2015 and 2016, on organic farms all over Manitoba and a couple in Saskatchewan. And so you can see that, so that blue line at 0.2% is sort of the cutoff for where we would say there starts to be problems below that. And you can see a number of farms are in that below 0.2 zone. We also have quite a few that are above. And if we want to get into a more nuanced recommendation, we can subdivide it into more than just, good and bad, below and above that blue line. but into some more specific zones of kind of medium high, very, very high. And there's more information on this in your instructions in that PDF document. So I won't go into any more detail on that right now, other than to say that there are specific recommendations that can be made based on how far above or below a particular field is above or below the line. We can also sort of divide it into zones this way, and how into where the data points tend to fall. In that red zone where soil phosphorus is really low, almost always the plant phosphorus will also be really low. In that green box where soil phosphorus is above around 10 parts per million, very consistently the plant phosphorus is okay, and even very good. In that yellow box is where the green manure bioassay results are the most useful, where soil phosphorus is probably rated as low, and in some cases the plant phosphorus is fine, and in other cases it's not. So if there's any doubt about whether it's useful to do the test or not, and you have some soil test results, you can kind of use that. You know, if they are in that 5 to 10 range, it is probably a good idea to do the test. If they're above 10, it's probably fine. If they're below, like 4 or below or lower, Olsen P, then it is wise to add a phosphorus source as soon as possible. Okay, so recommendations then. How will you, what will you recommend to the farmer? Phosphorus sources, manure and compost are still, I think, one of the best sources of phosphorus. It is fairly quickly available to the crop, and you might be surprised at how cost-effective it is to bring in manure, even from a considerable distance, compared to using some of the other commercial products. And I have some other information on that if any of you are interested, but that's not for today. Bringing in feed is an option for mixed farms. I know for organic farms it has to be organic feed, and so that can be a cost, but it can be an option. And then there's also mineral sources such as rock phosphate and some other like bone meal type products, and some other formulations that are out there. But again, those tend to be expensive. Application rate, a little can go a long way, but if soils are really very low in phosphorus, it may take a bit more of a substantial amount to sort of kickstart that system and get everything operating again. In terms of when is the best time to add phosphorus, we haven't actually tested this in a trial, but from our observations, we are now recommending that the phosphorus go on before a green manure or before the end of the forage phase to actually feed that nitrogen-fixing legume or forage. It helps support good nitrogen fixation in that stage of the crop, and it also allows for better management of any weeds that are stimulated to grow by the additional fertility that's been added. So that's our recommendation, but again, it'll depend on the farmer and their operation about when it's possible to actually add an input. Okay, I want to just talk again a little bit more about the plant nutrient uptake, only because we provide those numbers and they can be a little confusing on how to interpret them. So the plant nutrient uptake is calculated on that summary sheet. It's very simple math. It's just the total biomass multiplied by the amount, the concentration of each nutrient in the plant material. And it gives you an idea of how much that green manure crop actually got out of the soil. So for phosphorus, potassium, and sulfur, and any other nutrients that you would analyze, except for nitrogen, it gives you an idea of the nutrient supplying power of the soil. And we can sort of assume that the next crop will be able to get a similar amount out of the soil. Okay, we have some people saying that they can't see anything on their screen, so I'll just keep talking in general terms, and hopefully it shows up for all of you fairly soon. In the past, we have also been saying that the nitrogen uptake is a rough estimate of the nitrogen fixation by the green manure, as long as certain assumptions were met. And based on a reading of the literature over the last year, we've refined our method for estimating nitrogen fixation. And so we've added that to the tool. And so it's important to understand that, the nitrogen uptake, right here, in this example, it is 105 pounds per acre. That is what that green manure crop got out of the soil in its above ground biomass. That, if it was a pure legume, that would be a fairly good indication of the nitrogen fixation. But in these green manure mixtures, we're finding that really it is the legume biomass that really contributes and that affects the amount of nitrogen fixation. And we can no longer make assumptions about, if you have 50% or more or so, then it's fine. But rather, we need to actually look at the legume biomass. There are some relationships that's been established for grain legumes versus forage legumes, and from that we can calculate an estimate of the nitrogen fixation. So you'll see for this one it's only 45 pounds per acre. In this example, the legume was, I think, a little less than 50% of the total biomass, and so that accounts for the fairly large difference between the nitrogen uptake and the nitrogen fixation. So in the past, we have probably been overestimating the nitrogen fixation based on our previous methodology, and we hope that this new approach will be a lot more accurate. It uses data from all over temperate zones of the world, and they established some pretty good relationships. So that's what we're going to be using from now on. Okay, if I know at least one person is having issues seeing stuff on the screen, and so I hope that most of you are able to see what's going on, or that it catches up at least shortly after we move to a new slide. I'm coming back now to the big picture, to our organic management tool timeline revisited, and just want to go over again the importance of the farmer and agronomist relationship through this whole process. As you go through and are, for example, collecting samples and analyzing results, the more quickly you can get those results to the farmer, the better, so that they can make some decisions. And you might wonder, do they need all of this information, or are there certain things that are most useful to the farmer? And really, everything that goes onto the summary tab of that spreadsheet tool is really important for the farmer. And so that includes the total green manure biomass, the percent legume and percent weeds. Again, we don't really have, hard and fast target values for those, but it's important for them to know those. If the weed proportion is really high, it suggests there's a management issue going on there. Similarly, if the legume proportion is quite low, there could be some management issues in terms of what their mix is, if they're seeding a mix, or the weed management, that kind of thing. The nodulation is really important for the farmer to know if that legume has not nodulated. You need to ask that question: did you inoculate? And if so, how did you do it? What has gone wrong here? In terms of the legume not having active nodules on it. The soil, phosphorus, potassium, sulfur, pH, organic matter, all of those are really important information for the farmer as well. Soil nitrogen at this stage is really, I'll say, almost useless because it's measured at the peak of plant growth, and so it should be low. The only time I would actually have any concerns about a soil nitrogen value at this stage of soil sampling would be if you had a high nitrogen level in spite of that soil just having grown a whole crop. If you still have high soil nitrogen at that stage, then it suggests you have excess nitrogen in the system. The plant nutrient concentrations versus the critical values, that is also key information for the farmer, and it'll take a little bit of explanation to, basically like I explained to you, about what those mean. And that estimate of N fixation is really important. It's when you're discussing nitrogen fixation by green manures with farmers, it's also important to stress that that is not necessarily how much nitrogen will become available to the next crop, because there's lots of factors that affect how nitrogen cycles through all the systems. Okay, and as I sort of alluded to, each of these items opens some discussion on the management related to all of them, and they're not just numbers and results to be handed over to the farmer, but rather they're an opening point for going into some more detail on the farmer's management approaches. It's just like if you get a blood test done and the doctor just hands you the results and says, there you go, it's not really that helpful. Instead, you need to know how to deal with those results. And as I said, you want to report back to the farm as soon as possible because they may want to be actually changing their management. Okay, Greg is suggesting that if I go back and forth between the slides a little bit, they might update more quickly for all of you. So I wanted to talk a little bit about going beyond this tool and the types of things that you might want to be thinking about as you're visiting these farms, collecting data, making some observations. As I said earlier, this tool is not appropriate for sampling in other crops. Where's my pointer? There we go. This test is designed just to be used on legume-containing crops. And so if a farmer is like, well, kind of pushing for more data on a different crop, you can suggest that, if they are really wanting to, and if they are wanting to take care of the payment and everything, there are other tests, if they really want to, for specific crops at specific stages, and they can pursue that with a lab, and you can help them with that if you are able. But really, general observations are probably almost as helpful, except for in very specific situations, like I'll say like something like potatoes. But general observations will often tell you enough about the other crops and their fertility status. Also weeds, crop nutrition, crop management. All those general observations are extremely useful in interpreting the results that you get from this test and for helping the farmer work through what their management approach is going to be. And I also want to remind you that talking with the farmer is actually a very powerful diagnostic tool. Learning the field history, learning what their priorities are, that kind of thing is really important in interpreting all the results and developing recommendations for changes. Okay, so next steps for you, if you are wanting to get involved in using this tool, and especially if you're working with farmer clients, is to take a look at the completed example, especially if this is your first year on this. There's if you want to run through an exercise of actually entering some data, there's some sample data available on the very last page of the instructions document. And then I encourage you to get out there and collect your own data on farms. It's to that time of year now where sweet clover Green manures will be terminated anytime now, and so you need to look for chances to get out there and sample. And then I'd like to open it up for any questions. I see we have one from Arlen. The critical values are not appropriate for other crops, but the total nutrient uptake is still relevant and useful for a non-legume. Okay, yeah, that is a good point. It is the critical values that are not appropriate, that should not be applied to other crops. Just taking a crop sample and measuring the total nutrient concentration is useful, especially, I guess, if you do it, well, maybe I'll say only if you do it, if you have something to compare it to. So let's say you have two fields side by side, or a portion of a field where something, maybe you had a green manure on one part and summer fallow on another part, and then the next year you grow a crop, and to compare the nutrient uptake of those two crops can be useful. Or, for example, yeah, where manure was applied last year or not, or monitoring over time, one particular field over time. But just be aware that nutrient concentrations and uptake will vary according to crop and stage, and so you want to be really consistent with your sampling procedures on that so that you can actually compare over time or between fields. Any other questions? You'll note there's my email address, so you can contact me with any questions you have about the technical aspects of the tool or the interpretation of the results or the recommendations that you'd be giving to farmers. Feel free to get in touch with me. For those who are actually participating in the POGI nutrient management project, we're going to talk a little bit more about some of the housekeeping things of sampling, sample analysis and where to send that. And so some questions like that will go to Iris instead of to me. But yeah, if you have questions or concerns or feedback, or if you find something, I have checked through the spreadsheet fairly carefully to make sure there's no errors in the formulas and such, but it's possible that there's still some problems in there. So if you find anything that looks kind of dubious, please let me know, and we'll try to get it fixed up. Okay, so we have another question that has popped up. It's from James. Is there a way for the tools you've provided to account for the termination method? For example, estimates of how rapidly nutrients are available after mowing only, disking, grazing, etc. I will say that no, there is not really a way using these tools to account for the termination method. All of the information that this tool gives you is how much the crop, for example, the nutrient concentrations are, how well was that crop able to nourish itself from the soil that it was growing in. The nutrient uptake will give you an idea of how much was taken out of the soil. So, that amount is, for a green manure, it is being recycled back into the soil, but there are so many processes going on in that soil and with the moisture, temperature conditions, etc., that it's not a good approach to measuring what will become available in the following year. James, does that answer your question? It's not a very good answer, I know that, and we've struggled with that because it is the information that you get from this is incomplete. It doesn't give you a good sense of what will be available to the following crop, a very specific sense, I should say. But at the moment, it is the best that we have available. There are some calculators out there from the states that will give you an estimate of nitrogen availability based on the nitrogen concentration of the crop and the time elapsed since it was terminated. But again, they're just estimates. Basically, as more time goes by, more is available. The more you break up that green manure, or into little bits with tillage or mowing or whatever, the more you incorporate it into the soil, the quicker those nutrients will become available. Soil biological fertility or activity, rather, also plays in, and so. Yeah, that's the long rambling answer to to say that no, that there really isn't a way to account for that. Any other questions? So either you're all really smart and got it all just on the first try, or I did a terrible job of explaining everything and you're not even sure where to begin. And I'm hoping it's the first situation. Many of you were on this last year and so are kind of getting an update, and I hope that if you're new that it all made sense. And like I say, once you actually are using it, I find that especially if you're more of a hands-on learner, that things make a lot more sense as you actually use it, but also some additional questions do come up if you're actually using it. So.
Thanks, Joanne. I think it's more so the first case that you presented there. So thank you very much. That was a really great presentation. I thought it was very, very thorough and very informative. And I think it doesn't look like there are any more questions right now. Maybe there's just one more coming through. And so as it's coming through, sorry, Arlen, if I cut you off there, you can go ahead and ask your question. But I think what we'll do right now is say goodbye and thank you to those of you who have joined us this morning who are part of the agronomy training program, and encourage you to, if you have any questions, to ask Joanne Thiessen-Martins in the months to come. And so saying goodbye to some folks, but asking everyone who is part of the nutrient management program, if you're an agronomist with the nutrient management program, just to stay on for another 15 minutes or so, just so that we can go through a few technical details related to being part of this program. So I'll just take a few minutes now and let those people leave who are interested in leaving, and then wait for those agronomists just to just stay on. So thank you to everyone for joining this morning.
Looks like people want to stay on.
Yeah, it's true. It does look that way. Okay, well, then maybe let's just get going. So hello to all the agronomists who are in the nutrient management program. Thanks for taking the time to be part of the webinar this morning and for staying on for a little bit more. So again, this is Iris. I think you've heard from me before through emails and through webinars before. And so yes, what I wanted to do is just have the opportunity to give you some extra information, and also the opportunity, if you have any questions for us, this is a time when you can ask. I will start off by saying that maybe one thing that you've kind of noticed with this program is not only have you spoken to me in the past, but we've also had a few other people who have assisted with the program. So we've had Tiara from last year, we also had Luke, we also had Alice, and now I think you've been receiving some emails from Greg DeYoung, who you've been seeing on this webinar this morning. So what's happening there is that just kind of due to the way that the funding works with the project that we just have like a bit of a turnover with people who come and work with us who assist us with the program. So it's been really, really great for me to have people come and help out with the various aspects of the program, but you have noticed that there's been a few different people. We were trying to kind of keep some consistency there and trying to sharing information between people. So thank you for being patient with that. I apologize if there's been any information that's kind of slipped through the cracks because of that changeover of people. But for now, we have Greg DeYoung. It's been great to have him for, I think it's been a couple of weeks now, and you'll be hearing a little bit more from him in the weeks to come. And then, so what I'll do is I'll talk a little bit, and then I'll pass it over to Joanne. I wanted to go over how to submit samples. So I'm thinking that most of you have received your package in the mail. If you haven't received the package in the mail, it should be coming shortly. And so part of that package was we included some of the submitting forms that you need to use. So what I'm going to do right now is I'm just going to share my screen with you so we can go over that together. So it's just going to take me one moment to get that going. And it just looks like it's taking a second. But what I wanted to do is just share my screen and go over the submission. So for those of you who are new to the program, the lab that we work with is ANL Canada, and ANL Canada is a lab that's in Ontario. So all the samples that Joanne talked about this morning that you'll be taking from the field, those will be put into the bags that were provided to you and sent to ANL. And it just looks like my screen isn't quite... Okay, so my screen's not cooperating right now. Maybe it's just a buffer issue, I've been told. So, so I will, just trying to think if there's something to cover with you folks that doesn't require me to share a screen. No, maybe I'll pass it over to Joanne while my computer's just taking its time there to switch over if there's something Joanne wanted to mention.
Sure. Maybe what I'll do while we're waiting for that is to just give you a little bit of a glimpse into later in the season about what you're going to need to do for the other tool, which is the nutrient budgeting tool. And you can read, there's a little bit of an overview in it in the one document that was sent out, and there'll be more detailed information coming out later. But it's basically a tool that quantifies all the materials coming into a field and all the materials going out of a field, including nitrogen fixation coming in, and then it calculates the balance. So it's a way to identify any trends in nutrient depletion or accumulation that can be happening. And the way we do it is actually to calculate the budget over a complete rotation on one particular field, or maybe two on a farm. And so you will need to talk to the farmer about all of the crop history from that field or those two fields over the course of their complete rotation. That means perhaps they'll need to dig into some files, some paperwork, which organic farmers should have all of that anyway. It should just be a matter of finding it. But you'll want to know all the seeding rates, the crop yields of all the crops grown on that field over the past number of years. And then we use a couple different approaches to actually calculate how much of each nutrient was in those items. Ideally, we take some samples from the farm from the current year, some grain samples, or if they're growing hay, we would take a hay sample. Usually, I think the program pays for 3 samples to be taken and or to be sent in for analysis. So you would choose the 3 most important crops, let's say, in the rotation, the 3 that are grown in the biggest quantity. Send those in for analysis so we have an idea of what those materials actually have in terms of nutrient concentrations. And for other crops, or if you're repeating this for yeah, for other crops that you maybe can't have analyzed, we do have book values that can be used. But really, the crop nutrient concentrations do vary a lot. And so it is good if you can use, get some grain samples and hay samples from that particular farm itself, even if it's not from that particular field, but from a field that's similar, and get those analyzed. And then again, there's another spreadsheet tool that basically just does all the math. It adds up all the stuff that went in and all the stuff that went out. So you can see, for each year of the rotation, did you have a surplus or a deficit of each nutrient? So for example, in a green manure year, you should actually have a fairly large surplus of nitrogen because there's lots of nitrogen fixation going on that year. And in a year where a cash crop was grown, you will actually have a deficit of all those nutrients, most likely, as those nutrients were removed through the hay or the grain that was harvested. And then over a whole rotation, then you can see sort of the balance over time of what those nutrients are. And then it can help you and the farmer decide, okay, if we're on a trend of always, removing phosphorus, potassium, and sulfur because no inputs are ever added, then calculating that balance can give you an idea of how much needs to be added back in to bring it back into balance. So you'll need to, perhaps you want to talk with the farmer about that early on, that you will be collecting that information from them at some point. If they have time during the summer, you could actually collect their, information from previous years already sometime during the summer. The harvest amounts and the samples themselves from this year will need to just be collected later on after harvest. But you could get a start on that during the year if you are so inclined. Let's see. The other bit that I just wanted to mention while Iris is still trying to get her stuff up, I was going to show you this, but it's okay, we can just talk about it, is effective ways of working with farmers to integrate this new knowledge that they have about the results of these tests or your observations from their farms. And if you were in the program last year, you will have heard me talk about some of this before. But my observation has been that farmers are, I sometimes say farmers never do what you tell them to do, and that's not quite true. But they do have a tendency, which is good, I think, to take what you've told them and adapt it to their own situation. However, sometimes that adaptation is not always done in the most appropriate way. And so I think it's important for us as crop advisors to recognize that and to think of ways that we can actually provide them with information and with options to make good decisions. And so these diagnostic tools, like the green manure bioassay and the nutrient budgeting tool, as well as your own observations, can help to identify the root causes of problems. Then you can also, if they have something else that's going on the farm, or based on those results, you could explain, explain their system back to them, like what they've observed in terms of which crops do well or which ones don't, or issues that they've had with particular crops. With your agronomic knowledge, you can explain why those certain things might be happening. And then I think it's always important to provide several options for farmers that they can choose from. There's actually lots of literature on this talking about baskets of options or libraries of innovations where people can make their own choice of what fits well for their farm. And it's good if you can provide some theory and rationale for all of those so that they understand what the implications are of choosing any particular one of those options. And then also encourage them, and also do this yourself, but encourage them to really observe what has happened due to any changes they've made, and encourage them to then adapt whatever they tried to perhaps work better. A tendency can be to, I feel like there's kind of two types of people out there. Those who will keep trying the same thing over and over and over and hoping that next time it'll work without changing it at all. And the others are those that will try something once and then it didn't go perfectly, and so then they kind of toss it out. And I think a middle road there is a wise one where you do learn from your experiences and adapt whatever you tried so that it works better every time you try it. It's also important, I think, for crop advisors to always be giving balanced information with pros and cons, and also stressing that it is ultimately the farmer's decision, and there's other factors besides agronomic ones, such as financial ones and labor and risk and all those kinds of things that the farmer needs to decide. And one thing that a farmer pointed out to me just this spring that I thought was really interesting, someone who is just starting, I guess, to work more with outside sources of information, or with outside experts. They are very good at sourcing their own information from the internet and all sorts of places, but when you're actually working with a consultant or an agronomist, it takes a certain level of trust between the farmer and that agronomist. And for organic farmers, I would say that can be difficult because they have their experience with crop advisors has not always been positive, because it's just been, in the past in conventional, here, when you just apply this product, you spray this spray or whatever. And so this farmer recommended that maybe when you're first beginning to work with a client, it's wise to say only actually provide recommendations for one field until that trust relationship is built up a little bit more. So that's about all I had to share. I don't know, Iris, are you having any luck?
Thanks, Joanne. No, I'm not. The system's kicked me out a couple of times, so I'm hoping that everyone is able to hear everything that you said, Joanne, because I don't know if when the system kicked me out, if it also kind of disrupted the whole webinar. So I'm hoping everyone caught that. Maybe if you can just indicate through the chat if there was any disruptions there, you can let me know. So maybe I'll just open it up to just questions in general for both me and Joanne. I'll play around with this a little bit more and see if I can share my screen. All I wanted to do is pretty much just go over those forms, and also, just to go over the forms. So if I'm unable to do that, what I'll do is I'll send out an email to everyone with instructions on how the forms work and how to properly fill them out. Okay, thanks, James. You're indicating that it's all clear. But yeah, I guess I'll just open it up to questions for both me and Joanne for technical questions, and also some of the more detailed questions for Joanne. There's one question from Liz about where can I find the nutrient budget tool. So that is something that we'll be sharing with you later in the summer, but if you wanted to get that sooner, we can also send that out as well.
Yeah, I believe last year's version should be available on the Organic Agronomy Training Facebook page, but we are going to be updating it, in particular the nitrogen fixation estimates. So
okay, so maybe we could look at an old version just to get an idea, but not to use it, to wait for the newer version?
Right.
Okay.
Yeah, but it does give you an idea of all the information that you need to collect and that kind of thing, and just how it works.
I see we have a couple of people typing, so I'll just wait a few minutes. And also in that information package that was sent out to you, we also included a checklist. And with that checklist, what we're hoping to do is to help kind of keep you on track with the things that need to get done this summer. We recognize and see that there's actually quite a bit of information that you're collecting, tools that you're using. So hopefully with that checklist, you can use that to follow along.
Leaving conference.
Eldon.
Okay, so James is asking how many green manure bioassay samples does the program pay for and how many fields? So the way that the program is set up is that we do 2 fields per farm. So 2 fields would mean that it's, and the way that Joanne has described that the sampling method happens is you go to the field and you can collect a few samples, but ultimately you're putting all those samples together and taking the subsample. So it would be saying either 2 fields per farm or 2 samples per farm.
Okay, and Arlen's question about if a farmer has only 1 legume green manure field to sample, how about the for the second sampling, do the crops that followed last year's green manure? And yes, that is, I think, an approach that you can take. In my own experience with sampling crops following green manures, I was actually I was kind of sad when I analyzed my data because there was actually basically no relationship between how the green manure had done in terms of biomass and how the following crop did. So as a researcher, of course, you're always looking for significant effects. But the short story is there is not a terribly strong relationship, at least across a geographic scope, between how one particular green manure did and how the next crop did. If you can do any side-by-side comparisons, or if the farmer is committed to sort of to doing that sort of sampling over time, then it can be useful, I think. So that's something that you may want to discuss with the farmer, and yeah, decide whether to proceed with that. Okay, could you just review the soil testing protocol, the number of samples per field, composite sampling, and the testing depth? Sure, there's details on that in the instructions, but basically we're having you take both crop and soil samples at 5 points in the field, and I know that's probably a lower number of samples than that you would typically do for a soil sample, and so you are welcome to do more than that, but it does get to be time-consuming to do more. So we're suggesting that you take 5 samples per field. Well, I should say that you sample at 5 places in the field. At each spot, you would take 2 samples in the 0 to 6 inch depth, because there can be actually a surprising amount of variability just within a few inches. And if you are doing the deeper samples, which we do recommend if you have the time, you could take just one sample at each of the 5 sampling points. Yeah, take 1 sample from that 6 to 24 inch depth, or another deeper depth that works for you and your equipment. 6 to 24 is kind of the standard, but other options are okay. Then you would actually, as you're sampling, you would combine all of the samples taken from the 0 to 6 inch depth into one, and similarly all of the samples taken from the deeper depth into one. So what you're actually sending in is just a topsoil sample and a subsoil sample from each field. Michael, does that answer your question? Yep, okay, so James, to Arlen's question, would it perhaps be a good idea to suggest that the subsequent or non-green manure crop could be one of the crops included in the harvest sample taken later in the year? Yeah, that is a good suggestion, actually. Thanks, James, for that. So Liz is saying 16 is the standard for 160 acres, and so as you're sampling fields, especially if you are on a large field that's not too uniform, you may want to actually increase the number of soil samples that you take, at least in the surface layer. For a smaller field, it ends up being actually 10 cores from 5 locations, and so it does give you at least a general idea. I will say that this mid-season soil sampling probably shouldn't be considered a substitute for spring or fall soil sampling because nutrient levels in the soil can vary with the crop uptake and such. So if a farmer is wanting, to do some really good in-depth soil sampling, the best time to do that would be spring or fall, like typical soil sampling. Okay, and as related to Liz's comment that you'll need 12 cores to fill a bag, it depends on the diameter of your probe, of course, and you also, if you don't manage to fill the bag with your soil, it'll probably be okay. I've sometimes sent in some rather small soil samples and it is usually okay. So you don't need to feel like you need to take more just to fill the bag.
And so just continuing that conversation about the sampling, in the packages that you received, you will have the soil sampling bags there that were provided by AgVice, so those are the white bags. And you're also given brown paper bags, which you can use for your biomass. So for each farm, you will have received enough soil sampling bags to be able to do, you'll have 4 bags, so the 2 depths for each field, so 2 fields, and then at 2 depths, so you have 4 bags. And then you also have 2 plant sample bags. And you can take all of that and just put it into a box and use the shipping tag that you were given. So the shipping tag, that just allows you to just put it onto there, and you don't have to pay, you or the farmer don't have to pay for any of the shipping. It just goes directly to A&L. And I think I'm going to try to see what happens if I try to share my screen with you one more time. You can let me know if it works. Because I just wanted to go over the plant submitting. Yeah, I wanted to just go over that because we just had a couple of questions last year from the lab about that. So let's see. Is everyone able to see, Joanne, can you see that there's a soil form right now in A&L Canada labs?
No, I have, it looks like it's trying.
Okay. Okay.
It shows Organic Alberta soil submittal PDF, but oh, there it is. Now it showed up. Yeah.
Okay.
There it is.
Great. Thank you. So I just wanted to point out that in the information that you're included, where you have submitted by, please put your full name in there, like where it says agronomist info. And then for client grower, please put in all the farmer information, so you can put in their name. It's really important to, I just put in an example here of putting in an email over here. So for example, one of our agronomists is Arlen, so just a fake email there for him, so that you put in an email, and then that way that will be received by that agronomist. And it's really important to put the account number, so this way it gets charged to Organic Alberta. When it comes to the field ID, please put the same information on here that you're also using in the spreadsheet that Joanne provided. So Joanne provided the Green Manure Bioassay tool, and you're putting field names on there. If you can use the same field names on here, that would make things a lot clearer. And what I'm going to do, so we had sent paper documents for you for these forms, but I'll send you PDFs, just blank PDFs that are pre-checked like this, so that you can fill them out yourself. So you can put in all the information for yourself and for your farmer, but we'll just have it pre-checked like this. Because the way that the program works, there's very specific tests that we can run for the farmers. And then otherwise, yeah, so I just wanted to stress the importance of putting in the right information, because we receive the information and you receive the information, and sometimes the farmer gets it. And last year we just had a couple of situations where we couldn't quite track down the test results from the lab. And it would be the same for the plant form. So I will send everyone blank PDFs for people to fill out. And I think I will leave it at that for that aspect of it. And there's one more question from Dave, which looks like it goes to Joanne, and maybe we'll have that be our last question, unless there's any last-minute ones from anyone.
Sure. Okay. Iris, are you able to bring my presentation back up?
I will try.
Okay. So Dave is asking you whether we can explain the critical values again, especially when the analysis value is lower than the critical value. So I'm going to back up to, let's see if I can find, let's use, not that one. Let's use this slide. Can you all see it? It says interpretation at the top, plant nutrient concentration, phosphorus farm 2. I'll assume that you can see it unless you tell me otherwise. So get my pointer. Here we go. So the critical values in these green boxes, those are placed in there for your reference and the farmer's reference. If a plant nutrient concentration is below those critical values, then we suspect that there is a nutrient of that deficiency of that particular nutrient. And so that's what the critical values mean. As I said before in the presentation, it is a little more nuanced than that, as in, if you're way above or way below, then the management may be different, but these are the critical values at which we think that there could be a problem if you're below them. Okay, they're just pointing out that you need to click on the that you all individually need to click on the slide to actually be able to see it. So it's called interpretation in the list that's on the side, but there's a bunch of them that are called interpretation. There are 5 called interpretation in a row, and it's the last one of those 5 that we're looking at. There's one other one lower down, but yeah, there's the one that we want to be looking at. Hopefully you can all find that. And I'll keep explaining, and hopefully in a way that it makes sense even if you don't see it. So once we get our lab analysis back with the plant nutrient concentrations, we can enter the nitrogen, phosphorus, potassium, sulfur values into that spreadsheet, into that tool, the summary tab, and then compare them side by side with the critical values. So in the example that we have here, our phosphorus concentration in the analyzed sample in the white box is 0.12%, which is substantially lower than 0.2%, which is our critical value for phosphorus. That indicates to me that the plant is not able to get enough phosphorus out of the soil to actually support its proper growth. And so that is likely due to a deficiency of phosphorus in the soil or some problem with plant uptake. When we look at the other values for this particular example, we see that the others are all either similar to or greater than the critical values, and so that helps us narrow it down to actually being phosphorus that's the problem, and not just that the whole crop was kind of low in everything because it had grown really quickly or because it was quite a mature crop or something like that. Does that make sense? I see Dave has been having trouble with the screen not showing up. I hope you were able to kind of half listen during that. I hope that explanation was helpful. If not, then we'll take another run at it.
And also perhaps it's already 10:45 here in Alberta, so we've got kind of gone a little bit over our time. So maybe for the sake of not keeping people for too much longer, maybe if anyone has any further questions, please feel free to email myself or Joanne. And if it's a sort of question that we see might be kind of like a good general kind of FAQ question, then we'll share it with the rest of the group. So I think otherwise we'll wrap it up. Thank you very much to everyone for joining today. Sorry for some of the technical the technical failures, I guess, that we experienced in the last little bit here. But I think overall, I think it went well. So thank you, Joanne.
Well, thank you all for your attention, and I hope that the summer goes well for you, and we'll be talking again at another webinar later in the year. Yeah. So otherwise, so for next steps for everyone who's on the call here, you'll continue to hear from myself and from Greg. I'll send out the information about how to properly fill out the forms, and you'll just continue to hear through us through the summer as we support you through the program. So thank you very much, and have a great day.
Leaving conference.
Joanne Thiessen-Martins.
Leaving conference.
Iris.